Bio 181, Chapter 10, video on cell division, how cells divide. And now we're going to be looking at checkpoints that are going to regulate the cell cycle and determine if the cell actually goes through cellular division. So there are multiple checkpoints. We're going to learn about three. The first is between G1 in the cell cycle, gap 1, and the synthesis phase. The second is between G2, gap 2, and the M phase, which is for mitotic division. And later we'll find also for meiosis. That's the nuclear division. And then 3 is another checkpoint to see if the spindle fibers have accurately attached to the chromosomes. So number 1, G1S, commitment to divide. So remember, we've got a cell cycle here. The cell starts off, this is a baby cell, and then you're going to have two daughter cells over here on this side. Most of the phase here, let's just do this again, it won't hurt. So most of the phase, can you remember what it is called? All of it together, it is interphase. So most of it is interphase here in the cell cycle. So interphase consists of 3 parts. Okay. G1, S, and G2, right? So we learned that in the cell cycle video. So here we're looking at between G1 and S, we're doing a check. So there are enzymes in the cell that are going to go in. And at this point, before it allows it through, it's going to determine if we have the proper nutrients. So are there sufficient, minerals and vitamins and food, basically like sugars, growth factors, all of the things that are needed for the cell to divide. So nutrients, those are things that are necessary for the division and to make all of the enzymes. Growth factors, these are often hormones that are secreted by other cells, or the cell may secrete and receive itself. So there are signals that indicate the cell should divide. So it's looking for both of those things. The next checkpoint is between G2 and M. So that's looking primarily for DNA integrity. So between G2 and M, so M phase is here. Okay, this is the, we're doing mitosis in this chapter, so we'll call it the mitotic phase. Okay, this is the nuclear division. So the checkpoint here is going to look for the DNA integrity. So before it goes into the mitotic phase, it's going to check, was the DNA properly replicated? So it turns out that, when the enzymes copy the DNA, there can be errors that are made. And if there are mutations, which is just any change in the DNA from its original form, then it's going to block that progression. And the cell will either, for any of these, if they're not met, the cell will either go into a state of suspended animation, so it'll just stop moving forward, or it may go into programmed cell death, which is called apoptosis. So if it determines, oh no, we've made some mistakes in that DNA is no longer looking like healthy, there are lots of errors, then what we're going to do is we're going to go into programmed cell death. The last one is called the spindle checkpoint, and this is just to check that the chromosomes are properly attached to the spindle fibers so that when they divide, you're going to pull the sister chromatids apart and they're going to evenly distribute. And so that's going to be here in the middle of the mitotic phase. Growth factors. So I mentioned these earlier as one of the determinants in the checkpoint. So for growth factors, they are going to stimulate cell division. So here's a cell and it may, produce growth factors. Okay, maybe this is a cell that's going to create some hormones. Those will be secreted maybe through exocytosis here. right? So they're going to be dumped out into the surrounding fluid, and then those will be received by cells that have the appropriate receptor. So if it diffuses to a nearby cell and it's got the appropriate receptor, that could trigger this cell to undergo cell division. So, especially, so an example that I would like you to know is platelet-derived growth factors. So that's after you have a cut, any sort of tissue injury, those cells are going to secrete often platelet-derived growth factor that's going to indicate, wow, we need to rebuild the tissue in this area really quickly and heal this wound. The last slide for this section is on cancer, so a slightly different topic. This is unregulated cell division. So cancer is a very broad term. And so if a cell just starts dividing and the checkpoints are not adhered to, even if there's mutations in the DNA and the cell just divides and divides, it's like stuck on go, right? A lot of times it won't even fully make all of the components, so the cells may not be functional that are being produced. That's problematic. So that's cancer. Cancer is a very general term. There's lots of reasons that you can have cancer, from mutagens, physical toxins, to viruses, to genetic causes. And they can be caused by mutations specifically for genetic causes in oncogenes. So onco, this word group means cancer. And this is part of the DNA, right? So that's part of the DNA. So oncogenes, cancer, genes that have been associated with cancer. And then also there's a class of genes called tumor suppressor genes. They will actively look for cells that are, or they'll actively look for characteristics of cells that are dividing unregulated, and they will prevent that. So if there's a mutation in those genes, then we will stop that from occurring.